starting the recording. So now I'm going to jump in here. I'd like to welcome everybody to today's seminar, and it's my pleasure to introduce Evan Park. Evan actually did his bachelor's degree in our department in ENST and graduated in December 2017. And as an undergraduate, he was a member of the University of Maryland soil judging team that went to Illinois in 2017 and brought home the national trophy. He is also recipient of a couple of different scholarships. He got the Roy W. Simonson scholarship from the Mid-Atlantic Association of Professional Soil Scientists. And he also was a Golden Opportunities Scholar from the Soil Science Society of America, which paid for his all expenses trip to present a paper in Tampa. And then after he graduated, he went off to Alaska and worked for a year in the Tongass National Forest doing soil science work. That was quite a great experience. And then after a year there, decided to come back, and he began his MS degree in January of 2019. I will say he already has 2 published papers from work that he did during his undergraduate period, and hopefully we'll have several more coming out of his master's thesis. So without any further ado, I'll let Evan introduce his thesis topic today, and you can share your screen and jump right in. Oh, and I will say that I'm going to put the evaluation form link in the chat box, and I'll probably do it again at the end, but you should look there to find the link to do the evaluation form for Evan. So Evan, it's all yours.
Thank you very much, Dr. Norris. That's a very kind introduction. I'm delighted to be here to share today. And yeah, my educational journey has, Maryland has been just so important to me. I loved my work here as an undergrad, and I went to a temperate rainforest in Alaska after I graduated, but decided it wasn't wet enough. So I went and looked at some underwater soils, and that's what I'm going to talk about today. I'm going to share my thesis research, which was a study of subaqueous soils in South River, which is a subestuary on the western shore of Chesapeake Bay. And specifically, I'll be talking about the relationships between subaqueous landforms and soils, and how recognizing those relationships improves our understanding of subaqueous soil development and contributes to a more holistic understanding of estuarine ecosystems, so that we can manage and conserve them more effectively. So my thesis has 3 main chapters, and the primary work of my study is the investigation of subaqueous pedology and the effort to map the South River soils. This is what I'm mainly going to be focusing on today. I'll talk about the concept of a soil landform relationship model. I'll talk about the specific attributes of the model that I use to map in South River. I'll talk about its usefulness in predicting soil distribution and refinements that can be made based on what we learned. My second chapter is a detailed analysis of the mineralogy of South River soils. And the work of this chapter really sprouted organically from questions that were raised in the process of describing and classifying soils that I observed in South River. It developed into the type of thorough investigation that will probably appeal to about 5 mineralogy fanatics worldwide, but it hopefully does provide some insight that'll help improve subaqueous soil taxonomy, how we name and categorize these soils. Today, I'm going to weave some of those findings into the main story of mapping and model testing. And my third chapter focuses on soil halinity, which is the ocean-derived salt content of the soil. I conducted a review of current methods of measuring halinity and current systems for classifying halinity. The purpose of this chapter was to develop tools and terminology that describe soil halinity and that will aid subaqueous soil research. However, I won't be discussing this chapter today because it doesn't have the direct connections to the soil mapping work, which is what I really want to focus on and emphasize. Plus, of course, I have to leave some things out and keep everyone interested in the actual thesis. I don't want to give everything away in the trailer, so I want people to go and watch the movie. In today's presentation, as I said, I'm going to be focusing on the pedological work, the mapping work. So I'll start by reviewing a few fundamental concepts of subaqueous pedology and soil mapping, and then I'll talk about the process of applying and evaluating a soil landform relationship model. This involves drafting a soils map based on that model, and that happens before we conduct any field work. And then our field work is to collect and describe, characterize and classify these subaqueous soils in South River. And this is a part of the story where I'm going to tie in some of the mineralogical work because it applies to classifying the South River soils. And then this process includes comparing the observed soils to the expected soils, and then evaluating that comparison, seeing how accurately a soil landform relationship model can actually predict the real distribution of soils in the subestuary. So today I'll talk about the results of our evaluation, and then I'll talk about revisions that can be made to the conceptual model that will help us improve it and help us refine our ability to predict and map subaqueous soils. So before we dive into the South River study, allow me to just go over a little background information on subaqueous pedology and soil mapping. In the past 25 years or so, the field of subaqueous pedology has opened as soil scientists have forayed into shallow water environments with a pedological framework. And one of the pioneers of this venture was George Demas, who demonstrated that the sediments in shallow water environments undergo pedogenesis. They show evidence of soil development. Soil forming processes exist in these environments, and they create horizons distinguishable from the initial material. So these sediments meet the definition of soil. Demas also identified subaqueous soil forming factors, including things like bathymetry, flow regime, and parent material, which control the expression of soil characteristics. And identifying these factors was a vital development in subaqueous soil mapping because it facilitated the connection to one of the foundational concepts of soil mapping, which is the soil landscape paradigm. And this is the idea that certain environmental factors control the expression of soil characteristics and features. So if you understand those factors, you can infer how soils will be distributed on a landscape. Another way of saying it is the soil landscape paradigm tells us that if you know the landscapes, you know the soils. So identifying these subaqueous soil forming factors allows us to apply the soil landscape paradigm below the waterline. We can look at geomorphology and see that the water depth and the tidal range and other factors associated with a certain subaqueous landform will influence the soil that develops there. And so early subaqueous soil surveys describe these relationships between landforms and soils. And more recently, an illustrious alumnus of University of Maryland, Dr. Barrett Wessel, he went beyond a descriptive survey, and he used these relationships to develop a conceptual model for actually predicting the distribution of subaqueous soils. So here is Rhode River, and this is where Barrett developed the soil landform relationship model. He tested the model in West River, which is adjacent to Rhode, and now I am conducting a further test of his model in South River. So the Rhode River model, which Barrett, or Dr. Wessel, I'm going to get that wrong on today, but I want to call him Dr. Wessel because he worked hard for that degree. The model that Dr. Wessel described, it tells the story of a long, complex history of soil genesis. I feel as if I could start this slide by saying, once upon a time, the Chesapeake Bay, because it weaves this long, long story. Now, this slide visualizes the history of soil development, starting with point A on the left and moving through time to point F, which represents the present day. So starting with point A, this section of Chesapeake Bay is underlain by the Aquia and Nanjemoy formations, which are tertiary aged glauconite bearing marine deposits. This is the substrate of this part of the bay. Point B shows that Quaternary and Miocene materials were also deposited in the ancient marine environment of the mid-Atlantic coastal plain. Point C shows that during periods of low sea level, most recently in the late Pleistocene, the overlying materials were eroded and the tertiary sediments dewatered, consolidated, and underwent soil development. So distinctive subaerial pedogenic features such as iron oxide concentrations and alluvial clay horizons, they developed during this time. Point D shows that sea level later rose again, and about 8,000 years ago, the Chesapeake Bay took its modern shape. And when that happened, these tertiary paleosols were eroded by wave action. As that wave cutting front moved inland, shown in point E, suspended sediment settled out in the deepening subestuaries. Now, this process of erosion and deposition led to landforms and characteristic soil profiles. And that's what's represented here in point F, which again is what we see in the present day. Point 1 shows the shallow parts of Western Shore subestuaries, which are called the wave-cut platform landforms. And these are landforms where a Holocene age mantle overlies a truncated paleosol, which is identifiable by these relic features like argillic horizons or iron oxide concentrations. Now, point 2 here shows as the water deepens, we see these wave-built terrace landforms occur. And here, the sandy Holocene material settles out and forms these deep stratified deposits. And then finally, point 3 shows in the deepest parts of the subestuaries, the channel landforms, fine textured material finally settles out and it forms these deep fluid deposits. Now, these soils in the channel are usually dark because of organic matter enrichment and sulfide mineral enrichment as well. So this diagram shows the progression of platform, terrace, and channel landforms, which is the dominant concept in Western Shore subestuaries of Chesapeake Bay. There are other processes and other landforms that exist in these settings, and I'll discuss them shortly. But here I just want to clarify that this figure is not a comprehensive representation of the Rhode River model, but it does illustrate the main concepts. So now let me review the strategy that I used to evaluate the Rhode River model to see how well it actually predicts soil distribution. So first, I used the soil landform relationships that it described to create a draft soils map. And again, this was done before we ever set foot or set boat in the South River. Next, I sampled South River and then described and classified those soils. And then I compared the observed soils to the Rhode River model predictions and then analyzed that comparison to evaluate how accurate the predictions were. So first, the draft soils map. For this, I delineated landforms in South River based on the methodology of the Rhode River model. And then I determined the soil series that were associated with each landform. Wessel described 4 criteria for delineating landforms. First, proximity of a landform to other subaqueous or subaerial landforms. We used soil maps to see what landforms and soils were in the upland adjacent to the subestuary. So for example, if an emergent tidal marsh was mapped in the upland, we would delineate a submerged tidal marsh landform next to it in the subestuary. And to get a sense of the proximity between subaqueous landforms, we looked at the contours of the bathymetry. The contours also helped us look at Wessel's second criterion, which was the water depth where landforms occurred. The third criterion was the 3-dimensional shape of the landform, and the fourth was the slope and fetch of the landform. Now, fetch is the length of open water over which wind can blow and generate waves. So it directly relates to wave energy, and so by extension, it impacts the amount of scouring that will occur on the subestuary floor. So near the mouth of the subestuary, the fetch is high because wind can come up the bay. And so these are high energy waves and they cause more scouring. But near the head of the river, the fetch is low because the subestuary is narrow, and there's lots of windbreaks. So these 4 criteria that Wessel described, they're useful because they're easy to visualize, and they help us explain the depositional environment that we can expect to find in different parts of the subestuary. Understanding the depositional environment allows us to infer sediment dynamics and answer questions like, does the wave base touch the subestuary floor? Does it cause scouring? Will fine particles settle out? Or is the water energetic enough to keep them in suspension? So using Wessel's criteria, I mapped out the landforms in South River. And in addition to the platform, terrace, channel concept that I described earlier, our study site included a few more landforms from the Rhode River model. So, and as mentioned before, each of these landforms has a characteristic soil profile associated with it. There are submerged tidal marshes adjacent to emergent marshes where we expect to see histosols, that is organic soils, sometimes below a Holocene-aged coarse textured mantle of soil. There are also submerged shoal/saddle landforms, which are similar to the wave-cut platform, except that they exist farther from shore. These might have been islands that have eroded. They typically have profiles with a sandy Holocene mantle over a truncated paleosol. The tidal creeks follow the same platform, terrace, channel concept, but they're differentiated because they're smaller, narrower, less energetic areas. So we expect to see less paleosol erosion on the platform, and we expect to see finer textured Holocene material in the mantle on the platform and on the terrace as well. Finally, South River is deeper than Rhode River, so we delineated a deep channel landform. Technically, this is a novel aspect of our study site compared to the study site where the model was developed. We expect the soils in the deep channel to be similar to soils on other channel landforms, but it's possible that they could be finer because these are the deepest parts of the river, and ostensibly they would have the lowest water energy. So these characteristic soil profiles that I'm describing, they are well defined and classified soils. Wessel proposed soil series based on them, and one of the important things is that each landform type has one soil series associated with it. So this means that the landform map is essentially the draft soils map. What we have right here provides enough information to predict the specific soil that we can expect to find at each location in the subestuary. So since the Rhode River model tells us what soils to expect, the next step in evaluating the model is to sample, because sampling will reveal how closely the actual soils match what's expected. So we sampled South River in transects that ran shore to shore across contours so that the sampling points covered the gradient of high to low points in the subestuary. The sampling points were placed in the center of each landform unit that the transect line crossed. And each landform type was sampled in several locations so that we have several replicates of what soils on these landforms look like. So once the sampling strategy was determined, we set out in the boat to get a look at the soils. We were able to do some of the description work in the field while we were collecting soils, but we also made sure to take lots of notes so that when we were collating data later on, we could keep things straight. Made sure to take super helpful notes. It was hot out there though, so it felt good to fall in. Let's take a closer look at points from this transect as examples of the soils that we observed. So here is the bathymetric cross-section for this transect. You can see that the Holocene sandy material overlies the tertiary paleosol material in the shallower parts of the subestuary, and the fluid fine Holocene material dominates the channel. So we sampled here on the wave-cut platform, and the soil exhibited the expected profile of Holocene sandy material over tertiary material. Note the dull gray colors of the Holocene material and the brownish reddish colors of the tertiary material. Next, we sampled on the wave-built terrace, and this profile was dominated by Holocene sandy material, which extends much deeper here than it did on the platform. We don't see the characteristic colors of the tertiary material until we're nearly 2 meters into the profile. Finally, we also sampled in the channel. And here we were able to sample with a Macaulay auger because the sediments were soft. But a Macaulay auger only collects 1 meter of soil at a time. So this figure shows one soil profile. This is the first 100 centimeters, and then it continues here with 101. It goes to 200 centimeters. It's a bit unconventional the way this picture is laid out, but this is one soil profile. And what we see is that this soil was relatively uniform. It's 2 meters of dark, fluid, fine textured soil material. And as a matter of fact, in the Rhode River, the channel landforms were sampled to a depth of 5 meters, and it was still this fluid material all the way down. These soils react strongly to peroxide, and even leaving them out on the exam table for a few minutes led to oxidation and to color change. Sulfide mineral enrichment and the presence of hypersulfidic materials was commonplace in the soils that we observed in South River, not just in the channel landforms either, in the sandy Holocene material as well. So after finishing this process of collecting soils with the field work and describing the soils, I classified the soils so that I could go beyond just a simple conceptual comparison of the material types and actually conduct a detailed comparison. Classification was done according to keys to soil taxonomy, and this is where my questions about mineralogy began. So allow me to pause the explanation of the South River subaqueous soil pedology and let me tie in a little mineralogy interlude. And again, what I'm going to share here, it's kind of an aside because the mineralogy is a part of the thesis that's unto itself. But I do want to include the findings because they do relate to the classification of South River soils. So it's not a non sequitur. There is a connection to the main story that I'm trying to tell. South River is underlain by glauconite bearing materials. It's underlain by the Aquia Formation, which is shown here in green. And because of that, I expected the soils to be categorized in the glauconitic mineralogy family class. Now, glauconite is a mica mineral, so it's a phyllosilicate, it's a clay mineral, but it's often found in the form of these sand-sized pellets. So this morphology makes glauconite unique because it's an important mineral both in the sand fraction and in the clay fraction. When I looked into the current edition of Keys to Soil Taxonomy, I was somewhat surprised that the criteria for the glauconitic mineralogy class is simply 20% glauconite pellets by weight in the fine earth fraction. It focuses solely on the pellets and completely overlooks clay-sized glauconite. Now, the justification for this is that this criteria allows for glauconitic mineralogy to be identified in the field because glauconite pellets are observable to the naked eye. However, in practice, mineralogy work is most often done in the lab. So including clay-sized glauconite in the criteria for the family class, you know, it's pretty reasonable. It wouldn't be a big departure from how mineralogy is usually investigated and evaluated. So I chose to look at the clay mineralogy of the South River soils because if there's a significant amount of clay, or a significant amount of glauconite in the clay fraction, then there's a case to be made that the family class criteria should be revised. So first, I conducted grain counts to determine the amount of glauconite pellets. And I did this in selected samples that were representative of the different soil material types in South River. I wanted to see if any of these soils, if any of these material types satisfied the glauconitic mineralogy criteria as it's currently written. The results indicated that glauconite pellets were present in South River, but not enough to qualify any of the soils as glauconitic. I also looked at glauconite pellets, in terms of the material type, and the tertiary material did have around 20% glauconite pellets, but other material types that were present in the mineralogy control section, they kind of dragged down the overall glauconite proportion of these soils. So next, I conducted clay fraction analysis, and specifically it was X-ray diffraction analysis, which I used to estimate the amount of glauconite in the clay fraction of the representative samples. XRD is a semi-quantitative analysis that allows us to identify different minerals and estimate the relative proportions in a sample. So the way it works is that X-rays are beamed onto a sample and then they diffract at different angles depending on the crystal structure of the mineral that they hit. Now, each mineral has a unique diffraction pattern, and that can be seen in the scans that are produced by X-ray diffraction. And minerals that have similar diffraction patterns can be differentiated from one another by scanning a sample several times using different temperature treatments or cation saturation treatments, or also by changing the angle at which the X-rays are beamed towards the sample. This figure shows 4 scans conducted on one sample, and the sample is horizon 3 BGB from the soil profile from transect 3.7. And this was a sample that was representative of tertiary material. So the 4 scans represent 4 different temperature and cation saturation treatments that were done to the sample. Overall, this figure indicates that this sample has lots of mica, which is represented by these peaks here, just below 9 degrees. And the sample has little of any other minerals. There are really no other significant peaks. This peak on the right side of the graph is there are lots of different minerals that all have a peak there, so it's not descriptive of any particular mineral. So what we can see from this scan is that the sample is mostly mica. And glauconite is a mica mineral, but it can't be distinguished from other micas like illite or muscovite based on this scan. The crystal structures of individual mica minerals are very similar, so an additional scan where the X-rays are beamed at the sample at a very high angle is needed to identify glauconite. So the peak that differentiates mica minerals from one another is called the O6O peak. And again, all X-ray diffraction peaks are a result of a particular mineral's crystal structure. And the O6O peak is a reflection of the cation that occupies a location in the interior structure of mica minerals. So for illite and muscovite, which have aluminum cations in their interior, their O6O peaks are here, close to 62 degrees. For glauconite, which has iron in its interior structure, the O6O peak is close to 61 degrees. So this figure shows the diffraction patterns of several glauconite reference samples that were used to confirm the location of glauconite's O6O peak. And the peak on the right side of the figure, it's from a substance that's added to the samples to serve as an internal standard that helps to align the scans properly so that we're confident that these peaks exist where they really are supposed to exist, and it's not skewed one way or another because of an issue with the machine. So when I conducted high angle scans on clays, on the South River clays, I saw that they too had peaks close to 61 degrees by and large. This is the same as the glauconite reference scans. They're not as pronounced, they're a little more like humps, but the important thing is that they're focused here around 61 degrees, so we identify them as glauconite. And so from this, I concluded that the mica in the South River clays was glauconite. And so then returning to the initial clay scans, I estimated that the clays of South River were generally about 40 or 50% mica, which means that they're 40 to 50% glauconite. So when I added the glauconite from the clay fraction to the glauconite pellets, about half of the representative samples surpassed the threshold of 20% total glauconite or were within the margin of error. Now, even though this isn't exactly the same standard, according to Keith's soil taxonomy, 20% glauconite is essentially equivalent. It's the same amount of glauconite, it's just not all in pellet form. So therefore, I concluded that the samples that had 20% total glauconite or were within the margin of error, these samples should be classified in the glauconitic mineralogy family class. However, overall, our findings were too variable to conclude that South River soils are glauconitic, generally speaking. Now, the presence of the sand-sized glauconite pellets, especially in the tertiary materials, and the predominance of glauconite in the clay fractions, in addition to the underlying geology of the study area, all of this constitutes evidence that glauconite is the most descriptive mineral for the South River soils. And I found that the South River clays were approximately 50% glauconite across the board. It didn't vary by material type or landform. They were by and large 50% on average. So this implies that surpassing the threshold of 20% total glauconite would just be a matter of having enough clay in the soil. And that's something, that finding is something that can be incorporated into a soil landform relationship model. If a survey area is underlain by glauconite bearing materials, we should be alert to the potential presence of glauconite, especially in the fine textured soils. If the soils have a high clay content, it's very possible that they'll have a high glauconite content as well and might fall into this mineralogy class. Now, there may be some of you thinking, my 5 mineralogy fanatics might be out there thinking, why is Keys to Soil taxonomy so focused on pellets when there's evidence that clay-sized glauconite is a considerable part of these soils, and it should be taken into consideration? This is a great question. This is my question. But part of the answer to that is that out of 20,000 soil series that are described in the United States, 12 of them are classified as glauconitic. And 6 of those are the soil series that Wessel described or proposed in the Rhode River study. So glauconite is an important mineral in the mid-Atlantic region, but it's less relevant elsewhere. And so it just hasn't merited the time and attention of the writers of Keys to Soil taxonomy. And I mean, this is something that I care a lot about and have worked hard on, but I'm pretty sure it's not going to be part of Biden's first 100 days either. That doesn't mean, however, that glauconite is not important and that looking at this definition is unimportant, because glauconite, again, it is relevant in the mid-Atlantic region, and it's relevant to subaqueous soils. And subaqueous soil taxonomy is a young, developing body of knowledge. It's something that's being worked on right now. And so getting the definition of glauconitic mineralogy right, you know, developing a definition, a criteria that is really descriptive, that captures glauconite influenced soils, that's part of the present work that's going on to describe and classify subaqueous soils generally. So I think that the South River mineralogical work, it at the very least raises the issue that the definition of glauconitic mineralogy merits some critical thought. And I think it demonstrates that clay-sized glauconite ought to be taken into consideration, and that as subaqueous soil taxonomy develops, that's something that really should be considered, that should be studied, and determined if you know, that should be part of the definition of glauconitic mineralogy. So with that, I will conclude the mineralogy interlude, and I'll return to the Rhode River model evaluation. Again, all this mineralogy work is a chapter unto itself, and I discussed the implications for subaqueous soil taxonomy, but I wanted to include it because, again, it does connect to the pedological work. I was able to classify some soils as glauconitic, but it wasn't a general characteristic at South River. So that, you know, there is that connection to the mapping, to the model evaluation. But let us conclude the interlude and proceed with the model evaluation. And looking again at the strategy that I outlined earlier, we've already talked about creating the draft soils map, sampling the South River soils, describing and classifying them, talked at length about classifying the mineralogy. So now let's pick up the story with comparing our soil observations to the predictions of the Rhode River model. The South River survey was evaluated by comparing the observed soil at each sampling location to the modal soil predicted for that location based on the Rhode River model. The comparison was done using an ordinal scale with a decreasing order of fit. And in fact, we used 3 ordinal scales. First, we used the scale that Wessel used in his model testing in West River, which we called the original Rhode River scale. Second, we used the scale that Wessel recommended as a revision to his original scale. So that's the revised Rhode River scale. And third, we used a scale of our own design, which we called the South River scale. So each of these comparison scales has a decreasing order of fit, which means that a higher number indicates a closer match between the predicted soil and observed soil. So in the original Rhode River scale, 1 meant there was no meaningful similarity between the expected soil and the observed soil. 2 meant that the observed soil matched the parent materials of the expected soil. 3 meant that the observed soil matched the taxonomic subgroup of the expected soil. 4 meant that the observed soil was similar to the expected. And here, similar is a technical term that means the 2 soils shared most diagnostic features and land use interpretations. And finally, 5, a score of 5 indicated that the observed soil matched the expected soil series. And matching the series indicates basically a perfect match. We found almost exactly the soil that was predicted for that location. Now, in his discussion of the scoring, Wessel mentioned that this scale needed revision because some classes were too broad and others were so narrow that none of the observations fell in at all. They weren't used at all. So he suggested a revised scale that focused on diagnostic horizons and features and removed the emphasis on taxonomy and similarity. So in this revised scale, absence of expected features and presence of unexpected features are given equal weight. And so we also developed our own scoring scale because we wanted to create classes that meaningfully distinguish between different levels of resemblance between the observations and the predictions. And so our scale, and to achieve that end, our scale focused on material types. So in the South River scale, 1 means no meaningful similarities, just as it did for the 2 Rhode River scales. And for the next classes, we proposed a series of questions about material types. Within 100 centimeters, are there at least 10 centimeters of slightly fluid to very fluid material? Are there at least 10 centimeters of non-fluid material? And is there tertiary aged material? Is there paleosol? Now, if an observed soil has all 3 questions correct, it gets a score of 4. 2 out of 3 correct, score of 3. 1 out of 3, score of 2. Now, these are yes/no questions, but a correct answer changes depending on the expected soil. So for example, if we expect to find a characteristic wave-cut platform soil, one with non-fluid Holocene sands overlying tertiary material, and then the observed soil is just stratified Holocene sand, then the observed soil would get a score of 3. It's expected not to have any slightly fluid to very fluid material, and it has none, so that's a correct answer. It's expected to have non-fluid material, the Holocene sands, and it does, so that's a correct answer. But it's expected also to have tertiary aged material, and that's not present in the observation, so that's an incorrect answer. 2 out of 3 questions correct, that's a score of 3. Now, for soils that satisfied class 4, they were also considered for class 5, which was the observed soil is similar to the expected soil, and also it would be considered for class 6, a series match. Again, class 5 in the South River scale, it reintroduces this concept of similarity, which Wessel had dropped in his revised scale. And class 6 is defined in the same way as the high score from the other scales. And again, a series match is basically a perfect match between observation and expectation. And the reason that we focused on material type is because the Rhode River model talks a lot about material type. It doesn't describe taxonomy quite as much or with quite as much detail. So having, you know, a comparison scale with questions about taxonomy, it just wasn't quite a match to what the Rhode River model really focuses on, talks about. So that's why we wanted to create questions based on material types, questions that were akin to the focus of the Rhode River model itself. So here's an example of the South River scoring system at work. The top row lists all the soil series that were described in the Rhode River study. And each of the 52 South River soil observations was compared to all 7 of these possible soil series. The column on the left just lists a handful of soil observations as an example. The numbers filling out the graph are the scores for the observed soil at each transect point compared to each possible soil series. So for example, in this box, we use the South River scale to determine how closely the observed soil at transect 1.9 resembled the Dutchman Point soil series. It received a score of 3, which indicates that it did not match all of the soil material types expected for the Dutchman Point series. You know, maybe it had unexpected material present, or maybe it lacked some soil material that was expected for the Dutchman Point. But in this same way, each observed soil was compared to each possible soil series and then given a score. So then after this was completed, we went back and said, okay, we've compared this observation to all 7 of the possible series, but which soil should we compare it to? And we answered that question using the Rhode River model. The green boxes on this slide indicate the series that was mapped on the landform where a given sample was collected. In other words, the green boxes represent the expected soil for each sampling location based on the Rhode River model. So these scores were then gleaned from the larger comparison matrix because they're the ones that actually demonstrate the relationship between South River soils and the Rhode River model predictions. So here's how the individual scores fell out for the different comparison scales. The modal scores for each of these were different, but then each of the scales had slightly different classes. So the question is, what do these scores mean? Was the Rhode River model effective in predicting the distribution of soils in South River? And this was kind of a difficult thing to assess statistically, you know, to get a statistically significant answer to that question is kind of challenging. Really, we started with the most basic version of the question, which would be, is the Rhode River model better or more effective than randomly assigning the distribution of soils in South River? So to answer that question, is the Rhode River model better than random soil distribution? We started by aggregating all the individual scores for each comparison scale, yielding what we called the observed map score. So the observed map score is the sum of the 52 individual comparison scores that we determined by comparing each soil observation to the predicted soil for its location. The observed map score served as the measure of the Rhode River model's success in predicting soil distribution. So for the original Rhode River scale, the observed map score was 87. That was the sum of all the individual scores. For the revised Rhode River scale, it was 131. And for the South River scale, it was 175. So following Wessel's method, we conducted bootstrapping analysis to test the significance of these observed map scores and evaluate whether the Rhode River model actually helped make a useful soil map. You know, based on the South River scale, the observed map score was 175. So the question is, does that score reflect the fact that the Rhode River scale really gave us a useful framework for inferring soil distribution? Or could you get a score of 175 by just randomly throwing soils out onto a map, you know, without any sort of paradigm or model or framework behind it? So we tested the null hypothesis that the observed map score is not significantly different than a map score generated by comparing our observations to randomly selected soils. And, you know, that kind of means that if the comparison is to a random soil, the draft map, you know, the expectation is created randomly. It's not created based on any model. So to test this null hypothesis, we resampled our 52 soil observations. And this process of resampling that I'm about to describe, we did it independently for each of the 3 comparison scales because we had 3 observed map scores, so we wanted to test the significance of each one. So in the resampling, instead of comparing each observed soil to the soil expected for its sampling location based on the Rhode River model, each observed soil was compared to one of the 7 possible series selected randomly. So for example, the Rhode River model predicted that the Conti's Wharf soil would be present at the location of transect 1.8. So we used this score of 5 in aggregating our observed map score. But now the resampling randomly selects Muddy Creek as the quote unquote expected soil for the location of transect 1.8. So that means that Muddy Creek is the soil to which this observation will be compared in the resampling. And that means that we'll take this score of 4. Now, this process, this random resampling was done for each of the 52 soil observations. And then we aggregated all those scores to get a random map score. This process was then iterated 10,000 times so that we had a distribution of 10,000 random map scores. And following Wessel's method, the 95th percentile of these random map scores was chosen as our significance threshold. So this threshold and the observed map score were used to test the null hypothesis. If the observed map score was greater than the 95th percentile random map score, then we would reject the null hypothesis and conclude that the observed map score is significantly different than a randomly generated map score. This would imply that the Rhode River model is significantly better at predicting soil distribution compared to randomly assigning soils throughout the South River subestuary. So looking at the distribution of random map scores based on the original Rhode River scale, we can see that our observed map score is well beyond both the 95th percentile and 99th percentile of the random map scores. Again, looking at the distribution based on the revised Rhode River scale, we can see that our observed map score surpasses the 95th percentile, and it's out here close to the 99th percentile. And then finally, for the random map scores generated based on the South River scale, our observed map score is well beyond both the 95th and 99th percentile. So this result tells us that we can reject the null hypothesis and conclude that our observed map score is significantly different than the random map scores. And the implication of this is that the Rhode River model is better at predicting the distribution of soils than randomly assigning soils throughout the subestuary. So statistically, what we can say from the bootstrapping analysis is just the Rhode River model is better than random. It's better than, you know, throwing darts or you know, making a soil map blindfolded. But the statistical test doesn't capture what the Rhode River model is actually able to predict and what it misses out on. So to get a clearer sense of what the model did well and where the model broke down, let's look at these histograms again. So the modal score for the comparison scores from the South River scale was 4. And this score indicates that the observed soil exhibits the presence or absence of all 3 soil material types as expected. And so the fact that this is the modal score indicates that the Rhode River model is successfully predicting the distribution of soil material types in South River. And that's a really important step. You know, if we can understand the soil material types, that goes a long way towards understanding the soil properties and interpretations of those soils. You know, based on soil material type, we can understand if oyster leases, if oyster bars are going to succeed, or it helps us understand if moorings are going to be stable, or if they will, if they'll move around in fluid material or something like that. Another thing that we can see from the histograms is that none of the South River observations matched the soil series that was predicted for their location. And very few of them were even similar to the map soil, the predicted soil. And the main reason for this was that the soils that were described and the soil series that were proposed in the Rhode River study, none of them included hypersulfitic materials. And by contrast, hypersulfitic materials were commonplace they existed in the vast majority of the South River soil observations. And so this discrepancy is the main reason that we didn't have any series matches or even any similarities. Hypersulfitic materials are, they have important implications in these environments. They constitute potential acid sulfate soils. They can be hazardous, toxic to plants if they oxidize. So this is a place where the Rhode River model breaks down. It wasn't able to predict the distribution of hypersulfitic materials. And the South River study, it does provide some insight. We do have some understanding of why hypersulfitic materials are more abundant in South River. And so that's something that this study contributes to the soil landform relationship model. It's something that we can incorporate, and so that in future studies we'll be able to predict the distribution of those materials in other subaqueous settings. So the take-home message of this study, of my thesis work, is again, this was a test of the model, and the result is that the Rhode River model is useful for predicting the distribution of soils in Western Shore subestuaries of Chesapeake Bay. The observed map scores we found were far beyond the significant thresholds for all three comparison scales. So we can reject the null hypothesis, say that our observed map scores are different than random map scores. And with that, we can conclude that the Rhode River model does provide a useful framework for mapping subaqueous soils in these settings. The landform, the relationships between soils and landforms that were described in the Rhode River model, they were observed in South River. They existed in South River. The Rhode River model was really good at predicting the distribution of material types. And with that, the Rhode River model helped us understand the land use implications for these soils. Again, things like suitability for aquaculture, suitability for moorings and docks, even aquatic vegetation success. Those are all things that the Rhode River model helps us understand. However, the Rhode River model was imperfect because it failed to predict the distribution of hypersulfitic materials. So as this thesis work wraps up, what we've discovered is that we need to propose new soil series that will capture those hypersulfitic soils in South River. And these soil series will be incorporated into the revised South River soils map, which is a deliverable of my thesis. It's being completed right now. And again, we have some ideas of why hypersulfitic materials are more abundant in South River. It might have to do with the morphology of the subestuary. It can also be related to the amount of organic carbon in South River, or the amount of carbonate in South River compared to Rhode River. But as we zero in on an explanation of the hypersulfitic materials, that's something that we can incorporate into the soil landform relationship model. And at that point, it's no longer the Rhode River model, it's now the Rhode and South model, or the Western subestuary model, or something like that, some revised version. And the Rhode River model, it was good. It was really good at understanding and predicting soil material types. But now the South River study is finishing up. The Rhode River model won't ever exist again. And that's how it's supposed to work. As we gain new information, we're constantly revising and refining these conceptual models so that hopefully they come closer and closer to approximating reality. The revised South River soils map isn't just going to be based on the Rhode River model. It's also going to integrate what we've learned about hypersulfitic materials, about glauconite, about halinity, and other things as well. And so then future projects that map other subaqueous or other subestuaries of Chesapeake Bay, they're going to have a better framework to begin with, and that's going to help them produce better soil inventories, and ultimately it will help further advance this field of subaqueous pedology and subaqueous soil mapping. So the Rhode River model, it is a good foundation, and the South River model, it provides some revisions and improvements, again, to move us towards a better understanding of Chesapeake Bay soils. And this ultimately is with the goal of understanding this resource, of managing and conserving this incredible ecosystem. And so with that, I just want to thank my committee. I want to thank my advisor, Dr. Ravenhorst. I also want to give a big shout out to Dr. Barrett Wessel, without whom this project couldn't have happened. And there were many, many others who were just such a huge help. So I want to thank them. And if we have time, I would like to take questions.
Okay, thanks very much, Evan. We're pretty close on time, but maybe we can take a couple of questions. And if people need to peel off, that's fine. But we know you're not going to run to a class, but you might. If you have an obligation, that's fine. But we're glad to take a few questions here. So, and I will point out that I have placed into the chat box the link for the evaluation forms, so be sure to take a minute to do that. You can, anyone have a question, you can type it in here into the
I
chat.
had a question actually at why people are typing in questions, and that was, I was curious about the integration of these models for subaqueous soils into something like the web soil survey tools that would be broadly available. You know, I guess we don't no longer have the big thick books that like that some of us remember for the soil surveys, but what about that type of integration? Is that soon or far away, or kind of where do you see that?
Yeah, I think that is pretty close. And again, that's something that Barrett did for his Rhode River study, and so that's the goal of my study. And I think we need to, propose the soil series first based on our observations, and then once we kind of have that, once we have them in the format that Web Soil Survey uses, then we can take our revised map and kind of plug it in there.
Cool. So I see Dietrich actually on, and so maybe if you want to ask your question, Dietrich.
Okay. I guess I was just wondering, when you're bootstrapping for your statistical analysis, whether you're doing the bootstrapping with or without replacement in terms of like how you're picking the pool from the pool of the samples that it could be matched with.
Yeah. So we sampled with replacement. So the way that we did it was we randomly chose which of the 7 soil series each observation point should be compared to, and we did that with replacement.
Have you looked at what the results would be like if you did it without replacement?
No, that's not something that we've looked at yet. But could certainly be.
The thing that I was thinking about that I'm just puzzling in my mind, and I don't know whether it makes a difference or not, or sort of what the impact would be, would just sort of like you're sampling that you sampled according to sort of an assumed pattern, and I'm just wondering like how that might affect then the resampling if the pool that you're sampling from isn't constrained by the distribution from your actual sampling protocol.
Right. Yeah. No, that's a good question. And I think that, like, for example, we didn't have any soils that actually had a score of 6. So then in the resampling, the random scores, there were no scores of 6 that factored into that. So I think that it could inflate or it could shift the distribution of random scores higher.
Mm-hmm.
So I think that is a good question. But we looked at it the other way. And I think the reason for that was because we did still want to tie it to to the observed soils. Because if we did it without replacement, then it's just, you know, it isn't 7 soil series. It could be all 20,000 soil series. You know, at that point, we're not really comparing to what we observed and the way the observations matched any of the Rhode River soils. But that's not something that we've looked into. It is something we've talked about a little bit, and something that might still happen as the thesis finishes up.
Yeah.
Right. Maybe
Might be something like your brain about, Dietrich, because I know you're the pro.
I was just like, as I was listening to you talk, I was realizing that I made a little mistake in my thinking, in that what I think what I was really trying to think about wasn't necessarily replacement or not replacement, but like constraining the discovery proportions, like the expected discovery rate of the different you know, soil series that you're looking at. But that's diving into the weeds. We don't need to go there.
No, well, thank you for the question.
Yeah, for time, I think we'll ask one more question, and that question is: do severe events like hurricanes affect the stability of the soil? And if so, how much would they change?
They do, they can. But what we found in reviewing bathymetric surveys from, Barrett found surveys all the way back into the mid 19th century, and I looked at surveys starting in the early 20th century moving forward. And in, at 50 years or 100 or 150 years of bathymetric observations, there was very little change in the landforms in the soils. And part of the reason for this is kind of thinking about the history of this part of the world. The greatest period of erosion was early European settlement, where these landscapes, these uplands, were clear cut, and there was a lot of erosion. And so since then, it's been less pronounced, and we do see that these landforms are relatively stable. And even events like hurricanes, which stir up the water, which cause a lot of runoff, they don't significantly affect the bathymetry, and so maybe they would cause brief periods of increased deposition, of more mantles, more organic matter being brought in, but they're relatively minor. They're not things that would change the way the soils are classified or anything like that.
Gotcha. Interesting, really interesting work. It'll be interesting to see it applied in other regions. I was thinking of the West Coast as you spoke, and thinking about San Francisco Bay and how this would, how this translates. So really, very cool.
Yeah. You need a submarine for those soils, though.
Yeah, I guess that's fair. All right, well, I think we'll end there. And just a couple of reminders, please go ahead and remember to give some comments in the evaluation. There's a link there for the chat. Please remember to tune in next week at 1 o'clock, where we'll have our 4 poster sessions. They're more than lightning rounds, less than not a poster session exactly, but we'll have those. And with that, I think
is that Tuesday or Wednesday next?
That's Tuesday.
Tuesday, great.
Tuesday at 1. Yep, on reading day. All right, very good. Thanks again, Evan. Really interesting.
Well, thank you everyone for being here. And I've saved the chat, and I'll try to respond via email to questions that I couldn't get to. Thank you, everyone.
That's great. Yeah, absolutely. Very good. All right.